What we have shown, we compared with the general standardized therapy for the pancreatic cancer, that is gemcitabine and oxaliplatin. Compared to both, what we have seen, this licofilone actually have the much better effect than the standard therapy. Then I will quickly add a few points on the non-steroidal anti-inflammatory drugs. We know everybody takes these things, but one of the safety profile problem is the intestinal tract toxicity, that is the ulceration. Then the licofilone also developed to avoid this ulceration thing and also to block the both Cox-Lox pathways. Why NSAIDs we have to thinking in the pancreatic cancer? There is a observational data where it shows relative higher intake of the NSAIDs have a lower incidence of the pancreatic cancer. Then there is several other in vitro, in vivo studies to implicate that phenomena. But the problem with the NSAIDs in the typical drug development, efficacy is important, but if you have the side effects, then how to avoid the side effects. There are several ways. We have a very number of compounds which are substituted compounds, like either using the nitro or phosphor groups. This nitro group compounds as a first generation substitutes for the NSAIDs we selected because of its some of the PGE2 mimic actions in the gut, so that it will protect the gut without going into the ulceration, all these things when you apply this thing and retain the same efficacy. These are the typical examples: aspirin, indomethacin, like we have so many compounds we tested. Just an example, the NO aspirin, this is the nitric oxide releasing aspirin. We used the 2 dose levels again. Then we did the typical histology after a period of time. Then what we have seen, we have not found the dose dependent suppression, but even at 100, that is the 1000 ppm, it is a low dose. Still you see a statistically highly significant suppression of the pancreatic ductal adenocarcinoma. So generally the most of the people work in the NSAIDs, they know like if you reach after a certain point, you won't get any additional benefit. Actually it will protect. So what interesting observation with this NO releasing aspirin is actually it blocked quite a bit in the pancreatic ductal carcinoma. Actually some of the slides maybe in the treated ones I can show them. Lot of eosinophil region, that is what we see. But in the regular ICC, like it is fully occupied the cancerous tissue. So we went up into the additional analysis of this molecular markers. So that is typically, this is proliferation marker. Since it is a NSAID, typically it blocks the COX activity. So we analyze the COX expression and COX activity. We have seen some inhibition, but higher dose, when we have an agent, it is releasing the nitric oxide simultaneously, you will see the some of the not so beneficial effect. So dose optimization is critical with these substituted molecules. So with that, what I want to summarize this thing is this particular model in the pancreatic cancer, unlike the other typical models like a xenograft or other model, this one is a true human etiology. Then if you intervene with the early, that is the in situ carcinoma stage, you have a much better protective effect with the various agents what we are trying to develop. I think so far we have, whenever we look into the signaling, if you select the right marker, probably one can do target in this animal model and see changes in the biomarker and the efficacy, then taken into the phase 1 and phase 2 clinical trials. With that, I want to thank you, my colleagues, some of them who have spent a lot of time on this, developing and assessing the effect of these agents and the NIH funding to do this work. Thank you.
Okay, thank you, Dr. Rao.